Okay, well, welcome. Thank you all so much for joining one of the Shoal webinar series featuring zoos and aquaria from around the world doing meaningful freshwater conservation work. We run regular webinar slots here at Shoal, and we're really delighted to be running one featuring Monica, Brian, Michael, and Alex. This is an informal session and it's an opportunity for you to ask questions, and we're aiming for it to last about an hour and a half. So please do feel free to contribute and participate as much or as little as you're able to or comfortable to. There will be a chance again to ask questions to our brilliant panelists. If this is your first time joining a Shoal webinar, I'll briefly introduce who we are. Shoal is a freshwater conservation initiative, and we're working on the world's most threatened freshwater fish species around the world. One of our flagship projects is the 1000 Fishes Initiative. We're working to mobilise conservation action for 1,000 of the world's most threatened freshwater fish species by 2035. But we also work closely with zoos and aquaria as part of this mission for conservation. And that's given rise to the Shoal Alliance of Zoos and Aquaria. And we have representatives of the Shoal Alliance here today on the panel. So if you're interested in getting involved with the Shoal Alliance of Zoos and Aquaria, you're representing yourself, a zoo or an aquarium, and you'd like to learn more about contributions to freshwater conservation, please do reach out. We have a page on the Shoal website dedicated to the Alliance, and you can contact us at info@shoal.org. If you want to learn more, please sign up to our newsletter and follow our social media channels as well. We're active on there, and I'll have a link to the panelists' websites and socials as well that I will send around. So thank you so much. Our guests today are Monni Bohm she is the Freshwater Conservation Coordinator at the Global Centre for Species Survival / Indianapolis Zoo. We have Michael Chase, who is the Conservation Policy and Strategy Specialist at New Mexico Biopark Society and the Albuquerque Biopark Conservation Committee co-chair. We have Brian Zimmerman, Director of Conservation and Science at Bristol Zoological Society and the IUCN Freshwater Fish Specialist Group co-chair, as well as being the EAZA EP Committee Chair. And last but not least, we have Alex Cliffe, who is the Curator of Fish at the Zoological Society of London, the EP Coordinator for Cyprinodontidae, including Aphaniidae, and the EAZA a member of the EAZA Conservation Committee. So thank you all so much for joining us. I'm really, really excited to hear from you. Something that really struck me when organising this was that I initially asked our panellists to provide one image to demonstrate the work that they do. And that rule rapidly went out of the window because everyone had so much to say and they were so excited about what they had to share and the work that they were doing that I realised it was perhaps foolish to ask them to restrict their amazing work into one image. So we're going to kickstart the panel by hearing individually from each of our panelists and sharing a couple of images to really demonstrate what their work is. So the initial brief was to ask them to summarize their work in one image. We've been a little bit more lenient, so there's a couple of images per person, sorry. And I'm going to start off with Monni. I'd really love to hear more about what you do, and for those that are not familiar with your work, it would be really great to learn more about what you do. And I'll bring that photo up that you shared.
That's the first of about three. Yeah, I'm Monni Bohm, as Georgie said. I'm the Freshwater Conservation Coordinator at the Indianapolis Zoo's Global Center for Species Survival, which is a partnership between the IUCN Species Survival Commission and the Indianapolis Zoo. Essentially, we came into being probably around 5 years ago by now, and the role of our global center at the Indianapolis Zoo is to support the work of the different specialist groups of the IUCN Species Survival Commission. So I have the pleasure in my role to work really closely with Brian, for example, in the freshwater fish specialist group, as well as tons of other specialist groups that focus on freshwater organisms. Now, this is always my disclaimer. I originally always thought I was going to work on mammals and terrestrial ones, so you can boo me now if you want, and then we get this over and done with. But in my previous role at ZSL, the Zoological Society of London, that's when I sort of fell, not literally, but more metaphorically into freshwater by through working on biodiversity indicators. So how can we measure the state of biodiversity by looking at red list assessments for a ton of different species that make up a taxon group, and then we can say something about the threat in these groups. That's, I believe, probably 2010 is when I totally fell in love with what's on the screen right now, freshwater mussels. Mainly because I found myself sitting in a workshop with experts from the mollusk specialist group who were doing red list assessments for these critters. And I do remember that the experts preceded every single assessment by pulling up a picture of the species to show me how it looked entirely different from the one that we had previously assessed. And they would every single time asked me, can you see how it's totally different? And every single time I shook my head and I was like, no, but you're the expert, so I believe you. Needless to say, from there onwards, I became a little bit obsessed with freshwater mussels, even if they all look a little bit like rocks. They actually do really cool things. As you can see on the picture there, that little bit of the mantle that sticks out is actually mimicking a fish. This is how mussels of the genus Lampsilis get the host fish to come up to them so that they can give them a gill full of larvae that then can stick onto the gills of the fish and get A, a ride, and B, nutrition for completing the next stage of their life cycle. So I think that's one of the most fascinating adaptations. And so when I moved to Indianapolis, one of the things that I immediately kind of thought was going to be really awesome is that here I am, not quite in the hotspot of, or in a hotspot of freshwater mussel diversity, but sort of just slightly a little bit north of it, but that there would be ample opportunity to really get involved in some mussel conservation. And so very recently we started a partnership as one of the things that we're doing for freshwater, amongst others, with an organization, essentially a water quality bureau just up the river from us on the White River here in Indiana to support mussel propagation and restocking of mussels in our local rivers to improve water quality, and also to help some of the state species of concern to improve their populations. So that's kind of one of the things that we're doing, and happy to answer questions about mussels because they rock. To rein me in, Georgie, otherwise I would keep talking about mussels.
I think that's a good problem to have, and that's exactly why we asked you. So this is good. This is good. Fantastic. Okay. So thank you so much, Moni. I think mussels are certainly one of the species, well, not that mussels is a single species, but mussels more generally are a group of animals that are so readily overlooked. And when we think of freshwater conservation, I think it's fair to say that most people don't initially think of mussels when they think of freshwater and freshwater conservation. So as our next panelist, as an introduction, I'd like to introduce Michael Chase. Thank you so much. And I will share the image that you have sent forward to summarize the work that you do. Bear with me one moment and I will share that, but feel free to introduce yourself while I'm wrangling the technology.
Yeah, absolutely. Thank you, Georgie. So as Georgie said, my name is Michael Chase. I work for the New Mexico Biopark Society in Albuquerque, New Mexico, in the United States. Similar to Moni and her team at Indianapolis Zoo, we are also a Center for Species Survival in partnership with the IUCN Species Survival Commission. In my background, I like to say that I couldn't choose a taxa, so I just found a job that I'm able to work with all of them. So I focus a lot on policy work and conservation planning with the conservation planning specialist group. And I got pulled into a love for aquatics by working with Tim Lyons, who's the special advisor for the freshwater fish specialist group for reintroductions and ex situ conservation. And rapidly fell in love not only with freshwater mussels, but more specifically freshwater fish. And most recently, the work that we've been working on in this realm is back in June, we had a great opportunity that was funded through the SeaWorld Conservation Fund and a few other organizing sponsors to organize a ex situ conservation assessment workshop for freshwater fish across the globe. And what a ex situ conservation assessment workshop is we look at the different roles that ex situ, mainly focusing on zoos and aquariums or any sort of conservation work that's outside of native habitat, and look at what role from anything from insurance populations to assisted migration to mitigate climate change impacts to habitat. How can zoos and aquariums and our other partners that work in the ex situ space really help these species? And I thought this photo was one of the most kind of summarizing in that we were able to gather 27 experts from 8 different countries, including my three fellow panelists that we were lucky enough to have join us. And the main thing that I heard from all of these workshop participants is that we're starting to have unified conversations about species conservation and planning for freshwater fish across the globe. Um, on the red list, out of the species that are considered threatened on the red list, we were able to create ex situ assessments for about 10%, which doesn't seem like a lot in the grand scheme, but in one go was a pretty ambitious and successful effort. And we're very lucky to have a lot of support. So right now we are leading the post-workshop report. So getting all of these workshop discussions and expert input that happened into the workshop, and then the actual program recommendations that we have into a full workshop report that will then hopefully be published and made available most likely in the spring of next year, of which, again, all 3 of my co-panelists are helping on the report writing process, which I'm incredibly grateful for. So it's a super coming in and not being in the freshwater fish space for a long time, being able to see all of these experts that when I was describing it to my team that isn't on our conservation team, I was kind of explaining it of more of a gathering and discussion of friends, because all of these amazing people have worked together in some way or another across the board. And it was really great to start to have these conversations. And we're super thankful not only for Shoal's support, but also for Brian's support and the FFSG to continue to boost this work. And we're hopefully going to be able to have something sometime in the next several months that we'll be able to have a webinar and kind of give a bigger picture into what this process came about and what are some of the recommendations that we have for species across, I think, 6 different continents that we were able to look at, and a good number of species that critically need this work. I'm sure, as everyone on the call knows, freshwater fish have been largely overlooked, and being able to have a prioritized list of at least some of the species is what we're looking at as a really good starting point to hopefully encourage more of this work across the globe.
Thank you so much, Michael. That's fabulous. Thank you, Michael. I think something that's really interesting that comes up for me so far, even just from the 2 panelists that we've heard from so far, is that defining the contributions from zoos and aquaria to freshwater conservation, it is highly diverse, and it's not as clear cut as you would perhaps expect when you visit a zoo or an aquarium. You may think that the species that are being exhibited and are on display, really the conservation stops there, but actually it's far more reaching than that. Which leads me on nicely to Alex, and Alex's work with pupfish. So I will share the image now for Alex. Feel free to introduce yourself while I do so.
Yeah, sure. Hi guys, I'm Alex Cliffe. I'm the curator of fish at the Zoological Society of London. My journey with pupfish started in 2003, working closely with Brian at London Zoo Aquarium. And I never forget the first day when Brian showed me a group of fish that were extinct in the wild, and it completely blew me away that zoos were playing a role in safeguarding species that were extinct in the wild. And this was like 24 years ago, before any sort of formal process was put in place. And it really showed the foresight of ZSL and Brian and other colleagues in Vienna Zoo, Rotterdam, North America, where at one point there will be a framework to formalize threatened freshwater fish and manage them in a good way to then safeguard them, but also to look at, in the event of an opportunity of reintroduction or threats to current in situ populations, there was a framework within zoos and aquariums to deliver that. And that sets everything in motion, leading up to the formalization of the EAZA freshwater teleos tag. So EAZA is the European Association for Zoos and Aquaria, led by Brian, and that was a formal process of decision making on which species needed ex situ care. And then that linked into an opportunity where I was asked to oversee the pupfish EUP. So that's like an SSP equivalent in the States, of just making sure that we had enough populations of each of the species, working alongside universities, zoos, and aquariums, and increasing the representation. So we're not putting all of our eggs in one basket, so to speak. And then more recently, linking with the ex situ, then as Michael said, Michael and the team at Albuquerque hosted the International ex situ workshop, which sort of galvanized the work from an EAZA level. So it just really elevated these three species on an international level to really champion them for reintroduction potential. And one of the things I really engage with is the one plan approach. So we approached a couple of colleagues in Mexico that have already introduced a Mexican species called the tequila fish. And we wanted to utilize their skills, their approaches to communities, and their techniques to look at how we could introduce these three species within the guidance. And that really set things in motion to engage with local communities in Mexico. And it was pretty much a pivotal point where if the communities were not engaged with this, it could have completely folded the reintroduction potential. But the way we approached it was that we said, we've been looking after your fish for the last 25 years, and now it's time to bring them home. So it's been a real mind-blowing experience, because when we approached them in early last year, they went, come and meet us. We want to see you in person. Let's see if we like you. And if we like you, we will continue the project. So it was quite a nervous time, but we got there, and we felt like film stars. They were running up to us. The kids were hugging us, and farmers were embracing us, saying, we can't believe these fish are still alive. So a lot of farmers and people that remember the fish when they were in the wild, they thought they were gone forever. So connecting with these people that remember the fish and connecting with the people that have never seen the fish was quite mind-blowing, to the point now where the project for all three species is bigger than the fish. It's bringing back the well-being for the communities. It's developing school curriculums. They don't really have biology as part of the curriculum, so it's really enriching the next generation. Outreach programs, ecotourism, water management, optimizing agricultural practices, and it's been very kindly supported by a number of people, Reverse the Red, and also National Geographic, and more recently with Monni and her team at Indianapolis Zoo. So me and Moni were in Albuquerque with Brian and Michael and Tim, and we literally flew from the ex situ straight to the in situ. So it was a perfect marriage of that link from zoos and aquariums to actions in the field. And I think that really shows with that platform on a shoal level of linking both the in situ and ex situ. And we are in the best position that we've ever been with getting these fish back into nature. And there's a few things in the pipeline. You might have seen a small announcement of the Phoenix Species Project, which is $100 million for 100 species. And they wanted to nominate one of the three species as part of that project with co-targets of the other two. So it's the Veronicae, that's the Charcoal Palmer. And so going from literally a couple of aquariums holding them in 2003 to where we are now, it just shows, as Moni and Georgie were saying, it's the underdogs. It's these species that can make a tangible difference. And now it's a flagship species that was nominated within the Phoenix Fund, which is very excited. And Leonardo DiCaprio launched a video the other week, and he mentions Mexico in his promotional video. So it's amazing to be in this headspace now. I have had a lot of sleepless nights, but in a good way. You know, it's very exciting. So we are so close, but we'll certainly keep you guys updated in due course on the next steps. Thank you.
Fantastic. Thank you, Alex. I think that's a really beautiful summary, and I do have a specific soft spot for pupfish as well. So I'll try not to bring too much of my bias into to setting pupfish against mussels during this webinar. But it is really, really lovely to hear that work is developing so much, even from when I first visited Whipsnade when I started working at Shoal a couple of years ago now. It feels like so much has already changed for you, and that is so uplifting to see, especially because often when we have these kinds of conversations, one of the main things that always comes back is not having access to funds or resources, or not knowing where to start because there just isn't that capacity to start the work. So
Yeah, I think a big part of that process is the storytelling, you know, keeping that story going, and videos and engaging with communities is a very powerful incentive for getting that hook for funders and partners, really.
yeah, brilliant. I couldn't agree more. I think we'll have time to share a brief video that the team working on the pupfish have produced. So we'll hear from Brian, and then between going more into the panel discussion, then we'll share that. But Brian, feel free to introduce yourself. I will put
Can
up
you
the
can you show the fish picture first, Georgie?
definitely show the fish picture. That is something I
Thank
could.
you.
Oh, I'm
I
always
realize I
happy
didn't
to
tell
do.
you the order to put them in, but
It's
thank
okay.
you.
No worries.
Um,
Just let me know if you want to switch over.
okay, thank you. Hello, everybody. I think I know everybody on this call, or at least most of you. And it's great to be together again. Last time I saw some of you was in New Mexico, and at that fantastic workshop that Michael and Tim organized. So it's good to see you guys again. And thanks to Georgie and Shoal for asking me to participate in this webinar. It's always good to be with people that like fish and to talk about freshwater fish in particular. So the species probably won't be too much of a surprise for those of you who know me that I really want to focus on is called Valencia roberti. And this photograph here, which was taken by one of my project partners, Yanis Kapakos from the Hellenic Center for Marine Research, is taken in an aquarium. This is a group of Valencia roberti, and it's part of his PhD that he's literally just finished, that is focused entirely on the two species of Valencia that are found in Greece. And this species has been a huge part of my life for over 2 decades now. It started way back before the workshop in Mexico, way back before the regional species planning workshop in Vienna. This was something that was done at London Zoo back in, I think it was 2004. We had a small workshop of like-minded freshwater nerds from aquariums in Europe that were really concerned about not only the conservation of freshwater, but specifically about the lack of attention to European freshwater species. And when we had that workshop back in 2004, the fish that I've showed you here, Valencia roberti, or the Peloponnese killifish, didn't even exist in name because it was only formally described as a separate species in 2014. At the time, it was considered um, just another member of the species Valencia latornixi, which is the northern equivalent of this species, which is confined entirely to Western Greece. And in the case of this species, it was primarily confined to the Peloponnese, the peninsula in Greece. Uh, they're unique for many, many reasons, and the reason I was so fascinated by them and wanted to learn more about them was because of what I did know. They were not only um, reducing in number and being depleted in the wild, but they were also very unique. The genus Valencia is actually three species only found in Europe from a family, Valenciidae, only found in Europe. And the three species are together considered to be glacial relics. So they were fish that lived on the continent before the last ice age, and in small pockets in the case of the eastern populations in the Balkans and in the western population, the species there in the Iberian Peninsula. And they are still confined to those lowland springs, very close to the coast, but an entirely freshwater species. And they live in these low spring habitats close to the coast with very little current. But they are also real habitat specialists. So they need very good high quality water. They need water that is in a relatively narrow range of temperature. Um, Spring water that doesn't get too cold in the winter, but doesn't get too hot in the summertime either, which you can imagine in a place like Greece is not easy. And these tiny spring habitats are under huge amounts of pressure from humans mostly. So there's three main drivers of threat for these two species. The first one is climate change and the loss of water, particularly exacerbated by human overpumping of aquifers. The second one is pollution, where the springs get just regarded as dumping grounds and people have no regard for the species living in there. And that includes both domestic waste, but also runoff from agriculture. So pesticides and fertilizers running into the water. And the third one is the highly invasive mosquito fish, Gambusia holbrooki, which was widely introduced in Mediterranean Europe after World War II for malaria control. And that mosquito fish acts as a direct competitor to the Valencia species. And so the mosquito fish is a live bear, reproduces in very large numbers very rapidly, and quickly outcompetes this egg-laying species. And the reason I wanted to show this photograph in particular is because it shows something that I think only aquariums can do when we're talking about fish conservation, or at least they can do it much better than you can do it in the field. This species is found in it's small first of all, and it's found in densely aquatic um, plant heavy habitats. They're very difficult to study in the wild from a behavioral perspective, and it was only through aquarium work. In this case, it's the lab in the Hellenic Center for Marine Research's facility in Anavassos near Athens that Yanis was able to capture some of the most amazing things that we never knew about this fish. You can see that there's a fish in the right-hand side of the picture with a black dot on its side. That's a male. In fact, almost all the fish in this picture are males. They have the yellow underbelly and anal fin. The one just in front of it, though, is a female, and you can see they're much drabber in color. They don't have the barring, they don't have the yellow, and they don't have the black dot. And what Yanis was able to capture from his studies in an aquarium setting was just how the reproductive process works in this fish, how mate selection is chosen by the female. She has all of these males pursuing her, wanting her to release her eggs. And the males do this incredible sort of courtship competition swimming dance to try and get her attention. And this black dot appears on their side only during those nuptial periods. So they're trying to impress the females and get her to release her eggs. And so this was documented for the first time in the lab in an aquarium setting, and it would have been virtually impossible to try and catch this. And Yanis, in order to do this, he set up cameras focused on the study tanks and captured literally thousands of hours of footage and looked at what time of day the species breeds. And we learned that most of the reproduction happens at night, usually under the cover of darkness. We also learned that the preference for this species is to lay their eggs on the lower substrate. We'd always thought most of the eggs would be laid on floating vegetation, but in actual fact, we found that the bottom substrate is the most important habitat area, or the location for spawning and for egg depositing. And he also learned a lot about the interactions with this species and that invasive Gambusia, and both direct competition, direct predation, but also the levels of stress that the Valencia have and they endure when they are in the presence of the Gambusia, which was something that he was able to measure in the lab setting. So to me, it really shows the importance of aquariums in terms of not only direct study, but how those learnings from an aquarium population can really impact the conservation of the species in the wild, because we now know what the species needs in terms of egg laying. We now know how the species behaves in terms of its reproduction, when it reproduces, the time of day, and the time of year, which was something that wasn't quite so easy to do in the lab or in the field on its own. So that's why I chose this picture of this. And it's a critically endangered species. It was described in 2014 and instantly became assessed as critically endangered, as is the case with the sister species. So that's my choice.
Thank you so much, Brian. That's a fabulous choice. Again, I know as a fish enthusiast, I don't want to over egg the fish choices, but no, that's a really lovely story. And I think that it's something, again, that when you visit a zoo or an aquarium, you're not necessarily appreciating that so many lessons are being learned from the species that are being kept at the time, not just from the perspective of having them on display to educate the public, but we're educating ourselves constantly about what species need and what they do. And so many species we know so little about, even knowing that they exist, especially for freshwater fish. Their rate of description is far above any other vertebrate every year. And that's given rise to one of Scholl's projects, the New Species Report. So it's definitely something that blows my mind to appreciate just how much we still have to learn and how much good work there still is to do. So next up, I know that there have been more photos that you've very kindly shared. So we'll weave those into some of the panel discussions that we'll have. But Alex very kindly sent in a 2-minute video that I would really like to share, as I think it is another great summary and puts into perspective some of the community elements too of what Alex was saying. So I'm just going to bring that up before we move into the panel discussion.
I've been looking forward to this moment for about 25 years. I've been looking after these species for a quarter of a century. It's all good and well having them in zoos, but we needed to take it to the next step.
I can't believe that I am standing right here on the edge of what was once a spring that housed the Potosi pupfish.
We've been spending a lot of time with the local people. When I mentioned that we were holding them at ZSL for the last 20 years, they were thinking, wow, there is a chance for these species to come back.
Nosotros nos bañábamos allí con los pececitos. Ahí fue donde yo me enseñé a nadar.
Luego ya Livia nos poníamos a pescar los pescaditos.
Se acabaron porque se acabó el agua. Y nosotros nos daba mucho gusto ir a ver.
Yo tenía como 10 años cuando conocí a esos pescaditos. Eran muchos, se miraban muy bonitos de colores.
Me siento pues orgulloso y halagado para volver a recordar aquellos años, ¿verdad?
Yo estoy dispuesta a ayudar en lo que sea, en que vamos a hacer esto, yo le entro a todo. Y ahorita me siento bien emocionada.
Bueno, ya se están apropiando del proyecto, pero realmente ya. Y esto también ha permitido este sentido de identidad, de reconocerse o revalorarse o reconocer su propio espacio.
Vienen los nietos y hay que inculcarles que conozcan eso para que también le tengan amor.
Es que nunca la vi. Me emociona verla, conocerla, ver cómo es, de qué color es. Creo que es algo muy importante para la comunidad. O sea, no he visto ni una vez, nada más en video, que fue ahorita. Es una nueva especie que voy a ver, que voy a descubrir.
Aquí en la universidad tenemos el laboratorio de acuacultura, donde incluso se pueden guardar a las especies, se pueden resguardar a los ejemplares.
La verdad es que esto pues ha sobrepasado las expectativas. Todo el mundo quiere trabajar, quiere hacer algo, quiere traer a los Cyprinodontidae de vuelta al sur de Nuevo León.
Meeting the communities and working with our Mexican partners, it's completely changed my outlook on this project. It's more than the pupfish.
We talk a lot about the purpose of zoos and aquariums and botanic gardens, protecting nature and preserving biodiversity. This provides an example of how it should be done. The zoos are one step in a chain of getting and restoring wildlife to nature.
Thank you so much, Alex, for sharing that. I think it often audiovisual just summarizes things so much greater than a conversation or a sentiment can. So that's a really powerful summary. And although it was for one of the projects, I think the themings there really are a through line throughout all of the examples that have been shared by all of the panel. So we'll kickstart the panel discussion and we'll do that for the next 30 minutes or so. But if you have questions, we will come to them at the end. So please do feel free to put them in the chat and we will revisit them. Alex, I know we've just seen from your video, but I think in light of having just watched that and it being fresh in our minds, something I wanted to ask you about is for anyone who's watching, whether that's live or whether that's later as a recording. I know certainly for myself, when I wanted to work in conservation, I definitely underestimated how much of it was about people and how much of it was about it was almost a revelation that conservation is a social science as much as it is an ecological one. And I'm definitely getting that message from what you're saying too. Could you talk some more about that and how that has been a key component of the work that you've been doing on the pupfish?
Absolutely. I think the people are the key thing here. You know, fish can look after themselves. It's humans that are intervening and causing havoc with the habitats. You know, as Brian mentioned, some of them have been pre-glacial. So, you know, they've been around for quite a long time. So one thing that really stands out to me is legacy and succession. So making sure that all generations are engaged. Because, you know, from a hobbyist perspective, we're all fish geeks, but it's opening that up to the wider communities, the people that don't really know the fish. You know, they might have seen them in the spring, but how do they breed and how can they look after them? How can they safeguard the habitats? So it's making sure that multi-generational communities are engaged. And that's why we targeted meeting the farmers, the people that remember the fish, but also engaging with the schools and the children to make sure that there's a level of confidence that we can safeguard species for many, many generations to come. So I think that is an important factor. You wouldn't want to sort of bottleneck or be very short-sighted in long-term planning. You have to make sure you're in it for the long term, and you know, making sure that the communities are stewards of the fish for many years to come. We can just sit back and let them take ownership and put those fish back to nature. But from an ex situ perspective, you know, having the experience, you know, within, you know, EAZA and the ICAP in New Mexico, then linking to partners in Mexico, you know, there's so many things that I've learned how to approach communities. And it's completely put a curveball on our conservation action plan, because we had this framework. Yeah, we can do this from inception to delivery, but there's so many things I've learned of how to approach people and when and why. And it's making sure that you've got them on your side all the way through, and listening, making sure that they're listening, and also making sure that they're empowered and they've got a voice. So yeah, it's been a real big learning curve for me, but we're in a good space now. Yeah.
Thank you. That's really insightful, and I think it's something that increasingly is becoming more within the consciousness of conservation is so much of what we do is often obviously directed by how much funding there is, and that those project cycles are not, well, we can't let the perfect be the enemy of the good, and obviously they are good that we are having projects funded, but just thinking beyond those cycles, and that really seems to resonate from everything that you're saying.
Sure.
And Michael, I'd really like to tap into some of your insight, and I know that you kindly shared the work from the Ex Situ workshop, but you also shared a photo from fieldwork that you've been doing. Is that correct? Yeah.
Yes, yeah, and I'm happy
It'll
to
be
talk
great to
about
hear
that as
more
well.
about that side of things if you're happy to. I'm happy to share the photo too.
Absolutely, yeah. So, it's been touched on by all of the panel, but really focusing on this one plan approach and being able to integrate the ex situ and the in situ work into one unified plan, and making sure that the field experts are talking to the aquarists and vice versa. So this is a photo from Bitter Lake National Wildlife Refuge in the southeast of New Mexico. And this area is habitat for a federally protected and IUCN Red List endangered species of fish called Pecos gambusia, or Gambusia nobilis, which has recently been split into 3 species. And for me, it's been really amazing and insightful to work with Mitchie Tobler from St. Louis Zoo and University of Missouri, who is a close shoal partner and supporter. His team comes down to New Mexico every few months to kind of take populations for the species, and not only is it threatened by climate change and drought, but also hybridization from Gambusia affinis, which is the western mosquito fish. It's native to New Mexico, but it is just outcompeting the threatened species by a manifold. And it's been really kind of empowering for me to be able to work with his team. And just for scale of size, this species is endemic to these kind of sinkhole springs. So on the right side of that picture, there are 3 of my colleagues on the other side of the spring. And Bitter Lake is this very complex network of springs that were finding less and less of the species in these springs, and less springs that actually still have the species. But we, through Mitchie's team, just recently got a co-funding opportunity to be able to work on this species, both for eDNA work and potential translocation or assisted migration. And it's even as simple as reintroduction into a different spring that, from what we can tell, doesn't have any other species of fish in it, but it's within the same habitat. So it's been really kind of great, one, to get out of the office. I think it's really important to, go out and connect, and being able to actually work with the species that we spend so much time on meetings and over emails to be able to kind of plan around, but actually going out and being able to see these species always seems to give me kind of that reinvigoration and refill that passion cup to be able to go back into the office and be like, okay, it's worth it. We're making a difference. Let's keep going. Let's keep, fighting the good fight and putting in effort. But it's also been really great. As Alex said, that he's learned of being able to look at all of the different partners and see, as a zoo and a community-based organization that lives, or that's stationed pretty close to where the species is, how as a zoo and aquarium, can we improve our storytelling, and how can we actually highlight the species, and how do we get this species in front of our guests, and being able to tell that story and tell stories of decline And we try to focus, as many organizations are kind of shifting this way, to regional species and the species that we're the closest to and have the most impact, both in storytelling, but also being able to reintroduce this species. So it's been really fun to, leave the zoo and aquarium space and really start to get into the one plan approach and this unified aspect of protecting these species that need it badly.
Yeah, fantastic. Thank you, Michael. I think something that's really staggered me from working with Shoal and learning about the work that's happening on springfishes is I had vastly underestimated the sheer diversity, but also the level of threat that spring systems face globally. And intuitively, it does make sense, but I don't think that when we speak of freshwater again, that springs are necessarily the flagship habitat or environment that people think of. They're not always the most intuitive in comparison to a lake or a river, albeit those are spring fed. So thank you. That's brilliant. I want next to pass over to Monni again. I know that your enthusiasm spans far beyond fish, and so we're very grateful to have representation from other taxa. Otherwise, this would be a bit of a fish club. So Monni, I wanted to ask you, out of the species that you're keeping at the moment, what one really is your favorite, or one that you're especially excited about?
Yeah, sure. I'm happy here to wave the flag for all the other species. I literally don't mind having a good fish conversation, but I was invited specifically to do the other ones. Yeah, so can I just quickly add something to Michael's previous point? Because that again kind of shows a little bit of what zoos and aquariums also get involved in. Michael, you showed Bitter Lake. Obviously, literally just a couple of months, in fact, 2 weeks before we all went out to New Mexico to the workshop at Albuquerque Biopark that we already mentioned a few times, Indianapolis Zoo hosted a freshwater mollusk assessment workshop to help the mollusk specialist group and the IUCN to complete the global freshwater mollusk assessment. So essentially getting to a comprehensive assessment of the status of freshwater mollusks. And I've literally just spent the last few weeks cleaning up the spring snail assessments, and Bitter Lake came up quite a few times. So just seeing this, I was like, I don't know, should I shudder because it's all a little bit, it was a lot of work, or should I just go like, oh, look, there's Bitter Lake. But yeah, that just prompted me thinking about the spring snails again. And as you said, Georgie, that was not good news during that workshop, I think. I think once we see the final results published, we'll really see how incredibly threatened springs are, and how incredibly threatened also the spring snails are, obviously, that are dependent on them. Anyway, back to your actual question. Sorry, another shout out for the mollusks had to happen. So it goes without saying. At Indianapolis Zoo, we actually don't really hold so far that many freshwater species per se, but there's one particular project that is very dear to my heart because I think it again shows how zoos and aquariums really can have quite a big impact, and that is our us taking part in an initiative to bring back the hellbenders into rivers of Indiana. And that's where the hellbender photo hopefully comes up. Yes, yeah. So when Georgie asked me to find a photo for those, I thought, sure, I've taken photos of the hellbenders that we have in our tanks. It turns out, A, I'm not a great photographer, or my camera on my phone is really, really bad, but most of the photos look literally like somebody just had sneezed and something had happened. Interestingly, hellbenders are also referred to as snot otters, so maybe my photos were actually pretty realistic. But I picked that little one with a little pebble on his face, which is very cute, which is one of the adult hellbenders that we have here at the zoo. But really what the real magic of this project is that behind the scenes we have a facility where we rear hellbenders as part of a larger network of zoos and aquariums, all overseen by Purdue University, who are running this whole gig really, keep us all in line. But we essentially receive hellbender eggs, we raise them up to a certain size, and then they go back into one of the, unfortunately, very few rivers that have the right water quality still to hold hellbenders in Indiana. And so the picture on the right is actually me at my first ever hellbender release. I think I was literally just trying to convey good vibes to that individual in the net to make sure that it makes it and survives in the wild. But the really great thing about this is that clearly the people who spearheaded this at Purdue University realized that zoos and aquariums have the capacity to help with sort of the propagation, the raising need for the hellbenders, right? And so there's actually a number of zoos that we have here in Indiana and just across the border in Kentucky as well that are involved in this. And then when the hellbender releases happen, it's all just one big get together when the different zoos come together, as well as sometimes we have events, obviously for local participation and so on. The landowners are usually there who give us access to their lands to go across their land and put the hellbenders back. And it again, shows role of zoos and aquariums to provide the knowledge and space to get the animals to put them back out in the wild. It then also shows how important it is to link with obviously all the local people, the landowners, to have everybody on the same page that this is what we want to do. And then when you go out there and you just see the joy of it, and I think this is also what we saw in the pupfish video, the joy of it and how inspirational that feels. I think everybody gets something good back. And that then just reinforces us to do more of it, right? Go for the next iteration of this. And that's what really brings me joy. Also, obviously, again, when we go to zoos and aquariums and as visitors, we probably often kind of look for the exotic to see it. But ultimately, I think a really important role of zoos and aquariums is to help with their own local species. And so, again, the Hellbender project is really key here to kind of do something for our local biodiversity, as is the project working on the freshwater mussel propagation. That's kind of sort of upstream in Muncie, but we're helping to support that as well. And so we're trying to, while we might not have super many species, freshwater species here at the zoo itself, we're trying to really start finding projects that are sort of on the ground in our local community in Indiana that we can support to make a difference for freshwater.
Fantastic. Thank you, Monni. I think that's very counterintuitive to what a lot of people would think about zoos. The idea that you're actively seeking opportunities that aren't just about what's on display. And I know I keep laboring the point, but I think that it's something that often if I have conversations with people outside of conservation or outside of working in zoos and aquaria, that facet of the work that zoos and aquaria do is not something that's very much within the public knowledge, especially not on species that might look like snot.
I mean, it's like, I hope that's not what people's snot looks like, because that would be quite something. But I mean, they're also called old lasagna sides because they've got these little wavy little sides, which is also cute. They have the best names. But yeah, you're right. I mean, ultimately, obviously, we want to kind of show the stuff as well that we're doing in zoos and aquariums, and it's great to have that on display. But I think beyond that, there's a lot of stuff that zoos and aquariums are doing that may not even be that much in the public's sort of view, and that we maybe also can do better talking about and showing off. But we're working on getting this right. And I mean, I wouldn't mind a mussel exhibit, don't get me wrong, but that's in the future.
No, that's fantastic. Thank you, Monni. I think I'd be with you on having a mussel exhibit, but it might be a hard sell for the public. But people don't know what they want until they have it sometimes. You don't know what you're missing. Who knows? That could be the next big thing for zoos globally, for a mussel exhibit.
Listen, I'd much rather support a mussel propagation facility that puts mussels out in the wild and improves water quality than necessarily having them on display. Ultimately, that's also decisions that you sometimes have to take, right?
Yeah, there are always trade-offs to make, and I think that that was something that Brian was speaking about that really highlighted that for me with the decisions that we make in conservation have to be informed about things like habitat preference or time of year that species are reproducing, so that we can target specific areas, specific habitats, because we can't conserve everything. And similarly with resources, we often have to make that trade-off of public facing work versus work that's maybe hidden behind the scenes but is actually resulting in restoration. So there's always these trade-offs to make. Brian, I know that you shared some more photos before this session as well, and we didn't quite get round to them. And I'm very keen to hear more from you. I'm just going to review some of the images I have. Bear with me one second. So you shared 2 images of habitats.
Yes.
Would you be happy to share some more information about those, and I'll get them up on
Yeah,
the...
if you get them up. So these are 2 images showing the same location, but about 20 years apart from each other. And they are showing the main habitat, the best location, I would say, of Valencia roberti, which is in a place called Kiliadu, which is a small spring in the Mornos Delta region on mainland Western Greece. And I say that this was the best habitat because this was always the best location that we had when we first did our survey back in 2005. And pretty much every single year that the team went out to sample the site as part of an EU kind of big biomonitoring project, we would always reliably go to this spring because of the healthy populations of Valencia there. Now, it might look just a bit like a quagmire here in this picture, but there's a lot of floating pond vegetation you can see there. And this is where the spring outflow is. And it has had huge numbers of Valencia roberti compared to all of the other sites that we sampled. Now, I started by saying that the common name of this fish is the Peloponnese killifish, but the last time anybody saw one in the Peloponnese was when we surveyed in 2006 and we found one. So we're fairly sure that the species is now extirpated from that area. So this habitat here was a really, really important stronghold for the species. And one of the things that made it probably so good for them is there was a lack of Gambusia holbrooke. So the invasive alien species hadn't managed to colonize this area. And instead we had really strong, healthy populations of Valencia, as well as a number of other sympatric Greek endemic freshwater fish. But you can see here in the background, this is the spring is adjacent to agricultural land. And every year that we would go and visit, they would do crop rotation, and it would be a different crop growing there. Sometimes it was maize, sometimes it was wheat, sometimes it was cabbages. So we know that there was intense agricultural activity, and there is generally in this area. And almost all the water from the main Mornos River has already been diverted, I think, in the 50s to Athens for drinking water. So the amount of surface water in this area that's left is either used for the local communities or for agriculture. So this picture shows what it looked like in 2005, and this is from the first report that we drafted. The second picture is from June of this year, and I want to show you to illustrate the situation as it is now. And this was just sent to me by my colleagues in Greece about a month ago. So it shows the same spring, and you can see there's almost no water there. There is probably in the deepest spot less than 35 centimeters of water, and it's underneath that vegetation, that emergent vegetation that's there. So this is a real worry, and this is, what I would call a crisis for this species. The best habitat in the world for it looks like this. And to me, this illustrates the importance of regular monitoring and continuous monitoring. Even though we thought this habitat was safe for the species, we always went back thinking it was going to be the site that yielded the best population numbers, the greatest health of the individual fish. We almost got complacent. We knew a lot of the other habitats were degraded and there were lots of problems there, but we could always rely on this site. 20 years later, it looks like this. And it just goes to show that even when you think something's relatively secure, in a very short space of time, it can change. And I think what's really concerning for us is the fact that Greece had a very wet winter last year. So we would expect springs to be at their fullest capacity this year. And instead, it's looking like this. And what we don't know is why. It's counterintuitive. We would expect water levels to be higher, but instead they're lower. Is this a result of groundwater pumping at a greater rate for agriculture? Is it a result of the spring flow being diverted elsewhere upstream? Or is it just a climate change induced challenge that means there's just greater evaporation rates and in the habitat the spring can't support water replacement. So we're in the process now of trying to find out, first of all, what's going on, and secondly, to see whether or not we can go there and rescue some of these fish. It's logistically quite a complicated expedition to create and to get the right people there to go and check this site. But we're hopefully going to get funding from somewhere to try and send a team out there, do recce, understand exactly what's happening with the spring, and hopefully secure some more individuals of Valencia roberti to hold in the labs, the aquariums at HCMR. And the plan is ultimately, we'd already been thinking about this for a few years, looking at the IUCN guidelines for translocations, is to move some fish from this habitat to a much larger spring system back in the Peloponnese. And this is a location that the fish, at least in human history, were never known to exist in, but we know all the kind of basic habitat parameters are right. They seem to be like as though this other spring would be a good location. Some of the sympatric species are present there, and it might actually, due to the larger volume of water, be a much more secure habitat for Valencia. So this would be almost an assisted migration as part of a rescue mission, but we need to do that more detailed background study of the recipient site before we would undertake something like that. But yeah, again, it just goes to illustrate the fact that you think you understand a situation, and in a very short space of time with freshwater, it can often change very, very quickly.
Thank you, Brian. I think it's obviously a very sad point to have to make, but it's becoming an increasingly important one, an increasingly pressing one. And I think something that often strikes me about a lot of freshwater fish habitats, specifically for smaller species, is that unless you really know fish, you could assume that the habitat wasn't important, because a lot of the time to look at, they're quite unassuming. They can be small pools, they can be peripheral ditches by agriculture, like you say. They don't necessarily look grand in these big ways that you would anticipate a hotspot for a species to look. And so paying attention and having resources to make those observations about these species and where they're still able to take refuge is really invaluable for the future of their survival. And a lot of that capacity does sit within zoos and aquaria worldwide. I believe we've had a question, so I'm going to have a look at that now. For those on the panel, are you able to open up the Q&A section? I think there is one for Alex, and it's a great question from Steve, and it's fairly comprehensive. So Alex, if you're able to take a moment to read it.
Sure.
I will read it out for those who are not able to view it. But Steve Lockett says, Alex said, fish can look after themselves, which is an important message we constantly try to spread in our work on the tall masses of South and Southeast Asia. But for 150 years, these fish have been moved beyond native distribution ranges. And for the last 50 years, they have been bred in captivity and released in huge numbers into the wild, with no monitoring and often no consideration of native ranges. My question is, how do we, or should we even try to unpick the actions of the last century and a half to ensure we do things better in the future for these highly significant river gods? Half of the currently valid species are data deficient, by the way, and introductions undoubtedly impact on our ability to get them red listed more appropriately.
Well, it pretty much dovetails nicely into what Brian was saying, that, he needs to get out there and look at a potential translocation and assisted colonization of the Greek killifish. Before he does that, he needs to do an impact assessment of the new site. So, if you're following IUCN Red List IUCN reintroduction guidelines, then there's a framework there for sort of due diligence and responsible actions. So it's a real challenge. So, it depends on the situation, but, with something like this, a big cyprinid that is, I don't know, how big do they get? Is it like 1 meter, 1.5 meters long? They're a big fish. So a predatory fish like that, you would need to have some sort of due diligence of thinking, well, that's going to have a big impact on some of the native species that will be, significantly smaller. So it's a bit of a challenge to try and undo those actions. But moving forward, with reintroduction programs like the pupfish, we're quite lucky that these springs are currently dry. So we've got a bit of a blank canvas. So it's a pretty benign disease risk analysis and impact assessment. But that should be part of any sort of reintroduction or translocation, is to look at what impact that species could have. It could do more damage than good, putting it in another area that's not its native range. So, it's having a plan in place to maybe closely monitor those species to see how they do interact with the local species, whether that's fish or invertebrates or anything else that could have a detrimental impact on the existing biodiversity. So it's a real challenge. And then on the species that are data deficient, yes, we went through all of this at the workshop with Michael and Tim and colleagues. We know that there's some species that are on the brink of extinction, and we're trying to get IUCN red listing to catch up with experience in the field. So there's a bit of a chicken and egg with making decisions on taking action, but also having it in line with IUCN red listing. So that is a challenge in itself. But maybe I can open up to other colleagues on the panel to chip in as well.
I just wanted to add something to the IUCN red listing, which is kind of not the main point of the question. But as I mentioned beforehand, maybe quite a lot of times what limits us with the red listing is that we just don't have enough people involved in the process. And so what we were trying to do, not sure if we managed it, but we will keep trying over time, is, and I may mention now freshwater mussel, so I do apologize that I'm taking it away from the marcia briefly, to build some more capacity within essentially like a pool of potential assessors that wasn't previously tapped into. And I think we still have to do a lot more work to do, but through a program that's called EAZA Safe, which is the Association of Zoos and Aquariums, which is the North American, well, and some other parts of the world, but mainly North American Zoos and Aquariums Association. Their SAFE program stands for Saving Animals from Extinction. We've got a North American freshwater mussel program, and the workshop that I mentioned that was held in Indy was also doing, obviously, some freshwater mussel assessments, because, where's the point in not doing that? And we try to get some of our AZA colleagues involved in some of the red listing as well. I think we kind of need to figure out how we can get more people involved and make it clear that everybody can get involved in this process, to fill some of those data deficient gaps that you just talked about, Alex, as well. I also had something on, can we undo everything that we've done? And then I was like, it's been so hopeful so far, but then Brian talked about that spring, and can I take it down a notch even further to the non-hopeful bit? I sometimes think we can't necessarily undo everything that's been done, obviously, because the world's changed. I think what we can do is try and find the best possible solutions for some of the species that need our help. as well as trying to build towards something that gives us a sort of healthy freshwater ecosystem that then supports other species as well as ourselves. And so I think at least some of my personal motivation behind some of the mussel work, for example, as well as another project we recently supported, which was looking on after what kind of aquatic fungi, for example, are in our freshwater systems, because we literally don't know anything about those critters. Really, it all stems from what makes a healthy ecosystem, or an ecosystem that can sustain us all, including our species. And so maybe sometimes we can't undo everything. And that's obviously always, for conservationists, a very hard pill to swallow, I feel. But I think that also makes it so much more important that we really celebrate the wins that we can get. And I think particularly, like, for example, with the pupfish, there's something that's really tangible that we can essentially win at and make it happen, and then show it off as, look, it is possible if we really all pull together, because it's a team sport, right? Conservation is a team sport. And as Georgie already said, it's ultimately mainly a social science with some biology thrown in for good measure. It's kind of how I see it by now. But yeah. Anyway, I took us off the Marcia. I don't know why I did that, Steve. I love Marcias, so I'm sorry. You can have a go at me later.
Thank you, Monni. That's great. Steve has sent a follow-up to say thank you for your replies, and has given an example about Tor tumbra, a species with a range through Java and Indonesia across 5,000 kilometers to Yunnan, China, and in 6 countries. The species is data deficient and cannot get funding for conservation and has no governmental protection. But this fish is being bred in huge numbers in many of the countries for conservation. Huge complication for actually being able to understand it and to allow it to be red listed. So I don't know if anyone on the panel generally has comments around that, but I think it's a really a very powerful insight about some of the challenges that come with the red listing process and then conservation breeding.
Can I just kind of jump in on that, and also following up with what Moni said, because I think it's more difficult to undo the mistakes than to stop them from happening in the first place. And I think so many examples with freshwater and the challenges we have are because we're trying to undo mistakes and human error. And in the case of the Tor, years ago I was involved in a project in Nepal looking at Tor and Tor Puchtor in the Babai River. And the biggest problem there was the number of trout hatcheries, so that sport fishing could happen in the Terai Arc. These are for Westerners to come and go fly fishing in the Himalayas, and that was considered to be a big threat to the Tor species there and all the other species found in that river, because there were escapees, rainbow trout, getting into the river. And what I am always astounded by is the number of big projects, maybe well-meaning, that are related to breeding fish, usually for human consumption. You can think of tilapia and carp farms and everywhere around the world that are funded by the World Bank and by other development agencies, that end up going wrong and cost huge amounts of challenges for the local communities and for the native biodiversity that are there. And I feel like we just aren't learning the lessons that we need to learn fast enough to stop this from happening. And Steve mentioned in his message there that this is for conservation purposes. Well, who's decided that is the solution? Where is their legitimacy in saying that that's part of the solution? And I don't know what the answer is on a global scale, but certainly national frameworks and regional frameworks have to get much better at stopping these activities in the first place, because what we're going to end up with is just rivers filled with trout, lakes and ponds filled with carp, and springs filled with gambusia, and that's going to be it in the end. So I think there needs to be some kind of concerted action to stop those mistakes from happening in the first place.
Yeah,
Georgie, if I can build on what Brian said.
of course.
Yeah, I think it's Brian put it very well, and it's, one of the things in terms of local natural resource use and kind of this idea of, using these easy species that we know are easy to breed, that we can populate in huge numbers for consumption. But we've got to think back to long before human impacts, in which all of these species that we're trying to recover were likely in some way or another used for native and indigenous and local consumption and local use in one way or another. Like from Alex's video, this now critically or extinct in the wild species that they're working with, that it was used for local consumption. And I think it's the framing of, we know that what effective conservation looks like, and we know that if it works, we take it beyond not just preventing extinction, but to the point of full recovery, that it can benefit not just the ecosystem, but also, the thousands to potentially millions of people that rely on these species in different regions. And I think one of the challenges that we face in that is this reframing of, where are we putting our resources? Because those same international banks that are funding these giant fish farms could be funding our conservation efforts that inevitably will accomplish, when done effectively, both goals of species recovery and natural resource use by populations when managed in a co-beneficial way. And I think one of the things is the policy frameworks that we had, like Brian mentioned. I can only speak to policy here in the United States, but waters and like springs and wetlands are now only protected under federal law if they have a surface level connection to a permanent lake or river. So that puts huge stress and even more lack of funding into these kind of unrecognized, like these springs, like even small ponds, or even, very temporary bodies of water, or even puddles of water sometimes in different areas from heavy rain seasons, that don't have any sort of protection or recognition under different frameworks. And I'm sure there's different cases across the planet, but it's where we're putting our funding and how we're defining critical resources or recognized habitat that I see as one of the most challenging things that we have in the future.
Thank you, Michael. I think that's a really good observation, and that the co-benefits of developmental work also being nature positive is something that we really should try to empower and explore more and more, because we're we're running out of time to choose one or the other. I'm very cautious that we have just under 10 minutes left. If you're watching and you want to contribute more in terms of questions, do let us know. But I thought something that could be quite nice to wrap with, to try and sort of bring together everything we've discussed, is that a general theme of the whole webinar was talking about underdogs and underdogs within conservation, but more specifically looking about the contribution of zoos and aquaria to those freshwater underdog species. And I think throughout what has been covered today, it's been a real insight into the range of skills and resources and work that's happening towards those species contributed by zoos and aquaria, but also that there's a real need to communicate this work more broadly and get this kind of messaging across, not just about the species, but the potential work that can be done. I remember very distinctly 10, 15 years ago now, maybe, that there was that famous video of a sea turtle with a straw stuck up its nose. And that video, I think, did more for the plight of plastic pollution and single use than perhaps decades of study in literature publications, simply because it just got people right in the heart. And I think that there's still a lot of those stories within freshwater that just haven't happened yet. So we're really mobilizing and we're doing such brilliant work, and even demonstrated by the progression of the projects that we've discussed today, real progress is being made. But there are still stories that I think that once they capture the public, they will really skyrocket. And I wanted to ask each of the panel in around a minute, if you could describe one story that you think you would love to see aware in the public consciousness from the work that you do, what would you choose? And I know that that's a big semi-existential question, and I've thrown it on you at the very last minute, so take your time. But it's something that I ponder a lot, and I would be really interested to see what you all say. So whoever wants to go first, please feel free to be brave. Silence.
Moni, sure, this is your chance to talk about freshwater fungi.
I mean, we tried, we had a freshwater fungus in our Uproar Conservation Challenge, which we ran earlier this year at the Indianapolis Zoo, where we essentially kind of, in sort of like a bracket style competition, put different species against each other. And I was convinced that because the particular species that we chose was an Indiana species that was actually just recently described, that maybe I wasn't convinced it would do better. I thought it would do a little bit better, and it didn't. So I probably shouldn't talk about freshwater fungi. But another thing, and it's since I've gone down this route now, since you prompted me to at least mention the freshwater fungus, Brian, I keep going down that route. And I think it's not a specific story, but it's more a call that we need to be maybe a little bit more bolder and just shout a little bit louder, because in this Uproar Conservation Challenge were mainly underdog species. They weren't all freshwater, and the winner wasn't a freshwater species, but the winner was actually an invertebrate. And at the start of the challenge, everybody said, oh, invertebrates don't have a chance of winning this. What happened was that the team behind that species really got into the street, had a really good narrative of how their species was technically in the World Cup final, got people to vote for it, and went out in the news and on TV talking about that species. And with a lot of voting happening, in that species won. And I think maybe there's a story in there for all of us and a lesson in there for all of us to kind of go like, maybe we just haven't tried everything that we can do yet. As you said, we're doing a lot of stuff. We're writing a lot of good papers. Papers are not communication that everybody. I barely read a paper anymore. It's dry, it's boring. I will happily kind of admit that. So I think we're lagging behind some of the other stories, but I think we're catching up fast. And with some of the good news stories that are literally waiting in the wings that we can make a huge impact with, that are just around the corner. Comparatively, I think we'll turn this around a bit. So I see that the general public is interested in underdogs. I think we just don't give them the credit sometimes and hang our heads a little bit too easily in defeat that nobody cares.
Yeah, I completely agree, Moni. I think people don't always know that they're excited about something weird until you show it to them, and suddenly they're obsessed with the weirdest thing they've ever seen. I always love these conversations, and Moni, you did it a lot today, where what the average person would call cute is far removed from what most people in these types of conversations are very happy to call cute. And lots of things are cute, it just maybe not by the usual convention. Does anyone else from the panel have any kind of closing thoughts that they want to contribute before we wrap up?
Could I just say, somebody said early on that I think it was you, Georgie, that, conservation, when you first get into it, all of us that are, animal lovers, wildlife lovers, we do it because of animals, and we don't realize that it's so much about people. And I think the really successful stories have had quite often not the scientists behind it. They may have been the ones to kind of provide the facts, but it's actually a different sector that we really need to engage to try and raise the profile of these species. I mean, Alex is lucky he's got Leonardo DiCaprio behind his pupfish now. I mean, that's, who can do better than that? But, I am aware of a number of good examples where conservation success has happened for species that nobody knew about because they had a really good comms person behind it. The press team at London Zoo helped me with a project in Madagascar, which has carried on for such a long time, since 2013, and it keeps going and it keeps developing more and more every year. And I think there are good examples out there that we in the conservation community need to harness when we're trying to raise the profile of these species that really need attention. And I think that's something we're not very good at doing usually.
Yeah, I think the same thing, that this human connection really engages people that may not be necessarily interested in fish per se, but when you can see it can improve livelihoods and well-being and ecotourism and agriculture and education, then it's bigger than the fish. Actually, it's bigger than the fish themselves. An example of that with Omar Dominguez, who works in Mexico, a very good friend of ours. He was working with a fish species in Mexico where he approached the mayor and he wasn't interested in the fish. But when Omar approached the communities, they were really engaged. They wanted to do the work. So when the news got back to the mayor, he realized that the communities have his voters. So a week later, he was in the field with a net, posing with a camera, going, "I love fish," because he knew that the communities were voting for the next mayor, and he wanted to make sure he was in. So the power of communities is more powerful than any conservationist or any government body. So it can work top down or from the bottom up. It depends on how you approach it, but ultimately it's down to the communities and the local people that really drive the conservation forward.
I mean, I don't think I could have ended on a more succinct summary of what we've been chatting about for the last hour and a half, which has really flown by. So I will close there. But thank you, honestly, all of you, so much for joining and taking part and experimenting with a new format for Shoal webinars with me. I've really enjoyed learning from all of you, and it really is a blessing to learn about the projects that you're working on and see how they've progressed, even in the short time that I've been within the sector. Just seeing that is genuinely moving. And I know that we had more people sign up that may or may not have been able to meet today. So this will be recorded and posted onto the Shoal YouTube channel. If you have further questions because you've watched this at a later date, do feel free to reach out to info@shoal.org and we can pass on contact details to any of the panelists. And if you want to reach us more specifically about any shoal related activities, then please feel free to. Thank you all so much for your time, and we will see you in the next webinar.
Bye. Bye, everyone.
Thank you, Georgie.
Bye.
Thanks,
Thanks,
Georgie, for
Georgie.
organising. Bye.